Hello everyone, my name is Ashfaq Ahmed and I work in the field of structural biology and bioinformatics. Today we will cover the basic concept of protein design. I did not cover any video of this series, so this is a kind of first video of protein design. In this video we will talk about what actually protein design is, why we are doing it, how we are doing it, and what is the basic concept of this field. If you are not aware, the last Nobel Prize given in medicine was half of the Nobel Prize was given to Professor David Baker from University of Washington DC, and because of this concept or because of this area. So from now on, or from this year, uh, I would say that this field is growing day by day, and eventually it will reach to some higher level. So if you want to dig into this field, or if you want to have some information of this field, so I'm today we will discuss in the easiest way, the easiest possible way. I will try to convey the basic concept. This, after watching this lecture, you will be able to read paper and understand those papers substantially. So let's see what actually we will cover today. In today's lecture, we will cover protein design or de novo protein design. Okay, de novo means from the scratch, to design protein from the scratch. We will cover the current concept of proteins, we will cover diffusion and RF diffusion, and we will cover MPNN and protein design. Okay. Actually, what is protein design or what is protein de novo design? So, de novo design of proteins seeks to generate proteins with specified structural topologies or with some specified functional properties. For example, you are interested to design a protein with some specific topology of the structure or with some specific function you are interested in, and you cannot find a protein, you cannot find a peptide uh, in your natural system, then if you think why not to design my own protein, it should be interacting to this particular area, or it should be more stable to temperature, or maybe some other area you are looking for. So now you are able to design your protein. Okay. In this field, you are actually designing a protein or a peptide which binds with a given target. For example, you have a protein and you want to inhibit it or you want to activate it, whatever you say, with a certain peptide or with a certain protein. So you can design that partner for that protein. Okay. Design a protein or peptide that folds into particular topology. One of the problem in protein structure prediction is folding, but that problem is almost covered, or that problem is almost solved by the AlphaFold and also the other related like Chi1 boards and all those things, all those softwares or programs, I would say. So now it is easy to design a protein that and test it for a proper folding, like whether it will be fold or not. Roughly, if you ask me, so designing is very simple. Just type a sequence of amino acid and give it to the AlphaFold, and you will say, "Look, I'm designing," but it will be random. And if you start designing this way, it will take hell of ages, and you may not be successful for what you are looking for. Maybe that your random sequence maybe failed to fold correctly, or maybe some other problems. So that's why you have to first learn the basic concept that how to design. It's not a random process indeed. And third thing, With the help of this field, you can design a protein or peptide, again, certain catalytic sites, or what normally people call it active site. So there will be many more questions for which you need to design a protein. Let's say design a protein which is more stable, just like we are using in PCR tag polymerase. So tag polymerase is actually the specialty, or the the ability of tag polymerase is that it can handle high temperature up to 92 degrees centigrade, or maybe above this, because of those disulfide bonds. So now if you want to design your own structural tag, just like a protein which have the ability just like tag polymerase, and if you can, so then you can design and you can use it. Okay, so the you can opt this field for various different reasons. Okay, the current concept of proteins or enzymes, to what we call it, how to say, um, central dogma. So the central dogma, that is very simple. We have a DNA, and in the DNA that comes to the RNA or messenger RNA, and the messenger RNA then translated into a polypeptide chain which folds into a complete protein or a functional unit of protein. So this is the classical way by which a protein is coming into the natural system, from DNA to protein. So these transcription and translation, and overall we call it central dogma. So this is, I use the word classical because the design part is not a classical one. That is reverse, a kind of reverse of this step. So how it takes, or how it works. First of all, I want to introduce you to the diffusion. What actually is diffusion? Diffusions are actually, these are models of AI. Okay, you need to understand it, like AI has different models. So one of them, or one set of those models, are diffusion models, and they are called generative models. What are generative models? Those models to which you provide some data, you train those model on this and on certain data, and then you ask those model to generate this kind of thing for me, and those model will generate something for you. Okay, so if you are still confused, so you have Meta AI in your WhatsApp. If you are using WhatsApp, just give them some sentence like, I need a picture of dog playing with saucer ball in a green field. You will see the Meta AI will give you some kind of the same similar picture. Okay, so these all are the examples of diffusion generative model, because these models used by Meta, or used by some other programs, or DeepSeek, or ChatGPT, or whatever you say. So these are actually the trained models trained by those people who are who maintain these models. So they train these models with almost every kind of data, I would say. And then when a user like me or a user like you give them a prompt, what is a prompt? Basically, the sentence you provide to these models, just like if you ask this model, like, I need a picture of a dog in a green field. This is actually a prompt. So these model read those prompts and then use its training knowledge and gives you a picture. So these are called diffusion models, diffusion generative models, and they are like AI models. So diffusion models, they generate similar data to the data on which they are trained. For example, if you train a model on pictures, pictures of cat, pictures of dogs, pictures of tigers, whatever you say, pictures of many things, Okay, now you ask them to, I need a picture having three green dogs, two black cats, one Ferrari car, like this. Then these models are trained, they know, and they will give you a pictures according to your prompt. This is what I want to convey to you. Okay. Training in AI means provide a huge data and a model will learn. Okay. So once the model learn, then they will you provide them a prompt and they will give you this kind of picture. By the way, I got this picture from this source. Let me show you the source. Denoising diffusion probabilistic model. So it is actually a good practice to show the source. Just wait. Okay, actually it was here. Okay, so these diffusion models, when they are trained and you provide them a prompt, then they will denoising these prompt into some probabilistic picture, and therefore they are called denoising diffusion probabilistic models, DDPM. Okay, so I'm sure that by now you understand how you are generating pictures with those AI models and what actually those AI models are known. So they are known diffusion models or diffusion generative models. Why I discuss this? Because it has a great connection with protein design. Now, there is some concept to absorb. A trained AI diffusion model can generate a figure from the scratch. You understand it now. If you provide some prompt, the diffusion model will generate picture. That's okay. Okay. Then why not train these models on protein data or protein figures? You got my point? Okay. If these diffusion models are so powerful, which are, then why not we provide the data of protein structures, the data of protein figures, in other words, from the PDB or somewhere else. So these models will learn what protein look like, because for models, pictures are pictures, data is data, whether it's protein or whether it's cat or whether it's dog or whether it's human. Okay, so scientists tried like this way and they train these models on protein data. It's okay? Okay. What you expect in a protein data? Alpha helix, beta sheets, loops, the bigger things. There are so many other things like 310 loops and pi helix and blah blah, but I'm not going, just basically concentrate on the secondary structure elements. And we have basically three secondary structure elements, alpha helix, beta sheets, and loops. So if we train this data from the PDB, let's say from the PDB, the whole PDB data, so for the protein solved by X-ray crystallography, electron microscopy, or NMR, or maybe AlphaFold, then train all this data to a diffusion model. Now, if you ask the diffusion model that provide me picture of a protein having three alpha helices, two beta sheets, and connected by loops. This is a prompt. The model will provide you picture. Once again, the model will provide you picture. Just like in PNG, you can try this in Meta. I'm sure. I'm not sure whether the Meta models are trained on proteins or not, but they will provide you something similar. Okay. Now, fair enough. You understand what is diffusion, what are diffusion models, you understand how we can use these diffusion models in protein design. But remember, so far we just learned that we can generate the pictures of proteins based on the diffusion models. There is a program Rosetta Fold or Rosetta TTF Fold. Let me give you a brief overview of this program. If you are working in the field, so there were servers like Rosetta server and iTaser servers, and this Rosetta server actually the Rosetta server actually was using a Rosetta program name Rosetta. Then in Rosetta, they brought different versions, just like Rosetta TM for specified for transmembrane proteins. So what those programs were doing? Those programs were doing to predict the structure from the sequence and predict you the 3D structure. Okay. Then scientists thought that, okay, we got the pictures of protein from the prompt by the diffusion model. Now, how we can bring this picture into the reality? This was question number one. Question number two. For example, I have a protein. This is, let's say, this is my protein. For example, I am interested in a peptide or another protein which should binds here, and if it binds here, then it should not be another protein like this one, because this one may not enter into the group. I need a smaller protein. I need a certain topology. Let's say one alpha helices will be enough, and this alpha helices can come fair enough here. Okay, so these were the two questions. Question number one, we solved, we got the structure, we got a picture of a protein. Now, another question, how we can bring this picture into a real protein? One. Two, how we can generate protein of our own interested topology. Let's say I'm interested in a protein having one alpha helix, then a loop, and then another alpha helix. We call it a motif. Sorry. Okay, we call it a motif. One alpha helix, then a loop, and another alpha helix, like this. So then these were the two questions. So the program which we are using since a decade, and that was also developed by Baker Lab back probably 2010 or 9, or maybe in that era, because I started using from that era. So I used those, I mentioned those years, probably it's before than that, but it may not be, because before that there was a modeler for homology modeling by Sally Lab. So it was almost a decade ago that we are using, and you all are using these kinds of Rosetta server and all those things. Okay. Now, the scientists or the people of protein design, they just merge the diffusion with Rosetta TTA fold, because the Rosetta TTA fold suited with diffusion models. How? Rosetta TTA fold can generate a rigid backbone. For example, if you give them a picture, like this is a picture of my protein, and Rosetta TTF fold is able to provide you a rigid backbone of this picture. But whether this backbone will be folded or not, this is another question. Or whether it will be folded into a similar structure in the in vivo or in vitro, that is another question. But let it be for right now. Then this was success one, and they call it RF diffusion. If you read this word RF diffusion, that means Rosetta TTF fold, RF from here, and diffusion. So it's the combination of Rosetta TTF4 plus diffusion model. So Rosetta RF diffusion, I will use the word RF diffusion from now. So the RF diffusion can generate a rigid backbone of that picture designed by the diffusion models in first step. Okay. Remember, I use the word backbone. The backbone, I mean, in every amino acid, there is N terminus, then carbon alpha, then C terminus. So this is actually the backbone. I'm not talking about the R group. Okay. So Rosetta will only give you the backbone. The backbone of all the 20 amino acid is similar. They are different only in the R group. So the generation of backbone is not a big deal, I guess. Or it was not a big deal, I guess, because all the amino acids are similar. So you don't know that this backbone is of methionine or this backbone is of cytosine or alanine or this backbone is of leucine. So that is why they tried another method. They called it RF giant inpainting. What it means RF giant inpainting? For example, if I have this protein, this green one, if I have this protein, this block is a protein. Now, if I ask to, I need a picture of the protein like this. So I will provide this protein first to the diffusion model because the diffusion model is trained on the protein data. So the diffusion model knows what actually I'm asking. So the diffusion model will generate a protein of this shape. Okay, let's say I need this shape this way, a protein which should fit here. Let's say this is another protein which should fit here or like this probably. Okay, so I need this protein which should fit here. Now I will provide this protein to the diffusion model and will ask the diffusion model that to generate a protein which fits here. So the diffusion model will intent this area or will copy this area, just a kind of template, and will try to develop a picture of this protein, and that should fit here. Now I have this protein now, but it is in picture. Then the Rosetta part will generate a rigid backbone of this protein. Rigid backbone without, I would say, flexible part or without those R groups. Okay, now what is next? Then the Rosetta giant or N painting will convert the backbone into the C alpha. As I told you, the term RF diffusion actually means Rose TTF4 diffusion. That's it. Now I have this backbone for this protein. Then we need or we required another program to convert this backbone into some certain amino acid. But there is a challenge again because these certain amino acid cannot be blind. It will not be like if you ask me convert these 20 amino acid, this backbone of 20 amino acid into a real amino acid. So I will say alanine, leucine, threonine, etc., just randomly. It may not fold because for folding the combination of certain amino acid is necessary. If you don't believe, just give GS, 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 GS means glycine and S means serine, or just give GC, 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 glycine and cysteine. We call it GS or GC linkers. You just provide this to the alpha fold and ask them to fold it. It will fold it just like kind of wire. because these two amino acid in combination, they normally gives you a linker. If you want to uh, make a helix, but by mistakenly if you inserted a proline, so proline is a known helix breaker, it generates a kink in the helix. If you are interested in a straight helix, there should not be a proline in this helix. If there is, you see the structure or that part of the helix will be kink, folded, because this is a helix breaker. So actually I want to convey a concept to you that uh, predicting amino acid just randomly is not, you cannot solve the job. So there was another program, message passing neural network, MPNN, and that program actually it's a type of graph neural network architecture, and it learns, it is trained on the protein data and on the protein amino acid. So this program actually reads the backbone and predicts certain amino acids for that backbones, which should be or with a higher probability to fold correctly. For example, in this structure, this is the alpha helix and this is the beta sheet connected by a loop. Let's say, let's take it this way. Now, this MPNN will predict the sequence for that rigid structure based on this shape. The MPNN will try to keep the topology similar to that shape. But after all, it is a program. It can make a mistake, but at least it will try. So the MPNN will provide you a sequence of amino acid of the to that rigid structure. Now let's conclude the story because we have an amino acid. What we did, we provide a protein, we ask this, we provide this protein and we say I need a protein or peptide which should bind to this area. Program gave me this one, and this is a picture. Then the RF diffusion converts that picture into a rigid protein backbone, and apparently it looks like that it will fit it here, it will be fitted here. Now the MPNN will provide you 3, 4, 5, 6, 10 different sequences which are highly likely to be fitted and to be folded into this structure of protein. Okay. Now you have an amino acid sequence for that protein you designed against that certain catalytic type. What is next? Provide that sequence into the alpha fold along with your protein and then check whether they are binding or not, whether this sequence folds similar as I'm thinking or not. If yes, then your work is almost finished here. Just calculate some binding energies, blah blah blah. And now the next part is to bring this knowledge into the lab. So how you will bring this knowledge into the lab? You have a protein with proper amino acid sequence, just kind of back translate into the DNA sequence or into the messenger RNA sequence, and then order your primers, use the same structure as a template, same sequence as a template, and just set up a PCR experiment and start it from there, and just try to check whether this protein will be able to be folded correctly in the lab or not. If yes, then carry on your further more interesting experiments. Okay, now what is magic? There is some magic I discussed to you. In the conventional central dogma, we started from DNA messenger RNA into protein, but in protein design we started from protein picture actually. Okay, but I use the word protein. So from the protein, then back to amino acid, and then back to the nucleotide sequence. So that's why I use the classical central dogma term, Although I do not claim, I did not read this term in any research paper, but it's actually I use it, so I don't know whether it will be appropriate or not. So the protein design is exactly opposite to the classical central dogma. What are the application? You can design your enzymes, peptides with higher stability to what we call it protein engineering. You can enzyme peptides for therapeutics used as drugs. But personally, I'm thinking that the world is shifting to these things, and maybe in near future, we may not have a great luxury or so many projects for therapeutics because specifically uh, therapeutics in the field of organic molecules because they have so many problems like ADME absorption and detoxification and metabolism and excretion, blah blah. And so these proteins, at least they will be specific and they will provide more good solutions as compared to these drugs. The most interesting, fascinating thing which is coming for genetic diseases, you all are aware that in case of genetic diseases or hereditary genetic diseases, at times we have some missing protein or enzymes in the body, and because of that the patient receives some symptoms, some disability or some other thing. So why not to provide those enzymes from outside source if you can design it, and the fields will go on because it's really a starting. And if you want to dig into the field or to jump into the field, it's a time to be in the field, to start learning in the field, and you will drop yourself somewhere in the ocean. And good luck. This was the first video. I will plan to bring more and more videos of the field. So as I promised earlier, so this was just the first video of the field. Thank you so much. If you're liking my videos, please thumbs up and share it with your colleagues. Thank you so much. Bye bye.